Just a reminder here, we have exam 1 next Thursday, right? So we're going to do lecture exam 1 next Thursday at 12. So we're on track for that. And our first lab exam will be on the 19th. So make sure you're mentally ready for those. There we go. Continuing on with reproductive isolation today. Well, let's see. All right. Okay, so one of the issues. Okay, let's go ahead and back up here. So remember where we are. We have this. All right, so we have this kind of extreme range of what we consider to be evolutionary change. We've pretty much finished covering the microevolutionary mechanisms. So remember, I'm not going to write it again, but microevolution is change in traits or heritable traits or alleles, more technically, in a population, so that's within one species. And the mechanisms are natural selection, sexual selection, genetic drift, which includes population bottleneck or founder effects. We have mutation, change in the DNA code or alleles based on the mistakes made in the DNA copying. And then most recently is horizontal gene transfer, which is where genetic information from very, very different groups can be combined together. Those are all mechanisms for what we consider microevolution. Macroevolution, which we're going to probably touch on more this week and up to the test, is speciation, and that's what we call basically the allopatric speciation that we've talked about so far. So I think that in order to really think about what speciation means, like how do we know a new species has appeared? We have to have a kind of a definition of what a species is. There has to be sort of like okay, what do we use as kind of our guide for a population that's divided in some way? They've changed over time by microevolutionary mechanisms. How do we know species have happened? And we've already talked about that this takes probably greater than 10,000 generations. So it takes longer than we can sit and watch. So it probably takes longer than any one scientist or maybe even a generation of scientists could really, couldn't see it happening basically in one's lifetime. So how do we define when speciation happens? So this has been toyed with or touched on by several key folks. So species concepts have been around for several hundred years, and the first one that we can talk about really is Linnaeus, who invented the whole concept of genus to species, right? Like the species name, which is a genus and species combined together, was created by Linnaeus. So Linnaeus came up with, what could feasibly be the first definition of a species. And this is what we could call like a typological species. It's based on physical appearance. And remember, when I first gave you the shells, I had you kind of just group them based on what you thought were superficially related and which ones were not related. So Linnaeus could say something like, this is in the mid-1700s, species are as many as were created in the beginning by the infinite. And so obviously, I mean, it's a pretty vague definition, but if you kind of boil it down, Linnaeus is basically saying all the species were created, and essentially his job, and a scientist's job, was to kind of categorize and describe species as they were created or spontaneously appeared or what have you. So, what I want to point out is this is the definition for species, but it's not really scientific, it's not really testable. It's very subjective, right? So species here is a very subjective term. It's kind of based on the judgment call of the scientist to determine whether individual groups were similar or different enough to be considered the same or different species. So fast forward almost 100 years, or basically exactly 100 years, and Darwin gives a definition for species. And I guess the reason why I'm contrasting these is it really reflects kind of the change in the attitudes of the time. That there's an attitudinal change that happens here. And Darwin's what we could call like a nominal species. Species are individuals that are artificially grouped into species by scientists. And here's Darwin's quote. I look at the term species as one arbitrarily given for the sake of convenience to a set of individuals closely resembling each other. So, if you kind of think about it, both of these definitions are kind of saying the same thing. Scientists basically group species on superficial or similarities and differences. Darwin's just giving more of a humanistic aspect of it, basically saying that species is a human concept. It's not a natural concept. So so either way, Darwin's species definition was also not testable, right? Two different groups might come up with very different species classifications based on what they used as their traits or what their attitudes were, right? Okay. Fast forward another 100 years, one more 100 years, to the 1950s. So this idea of species concepts has taken a couple hundred years to kind of really, get to a point where it's useful. And Ernst Mayr, who was a bird biologist, here's Ernst Mayr's definition. And this is the species concept that is still used as the basic reference today. The biological species concept. Species are groups of interbreeding natural populations that are reproductively isolated from other such groups. And so, what Ernst Mayr's definition is based on is if you have one or more groups.
It's the construction.
Oh, okay. I thought that was our fuel. Anyway, if you have a population or multiple populations that are able to interbreed and produce fertile offspring, so that's what the definition of biological species is based on, is if individuals could reproduce and generate offspring that are fertile and are able to reproduce, they're considered to be members of the same species. And if there is any barrier to reproduction, natural barrier, then they're considered separate species. And so why this is important is this is a testable mechanism, right? You could, if you were curious if two populations were members of the same species, you could either look for hybrids or you could try to generate hybrids in the laboratory. But the one thing I want to point out that's a key phrase here that is often overlooked by folks is natural populations. You have to consider natural factors and natural populations. We can't use artificial environments or artificial breeding as a indicator that populations are the same species or different species. So we've got three species concepts. Obviously, Linnaeus and Darwin species concepts are very similar insofar as they're subjective, and Linnaeus was focused in on kind of more of a theistic definition of species. Darwin was focusing more on a humanistic definition of species, and Mayr is on a scientific definition of species, meaning that it's testable. Right? Sound good? Okay, so let's go ahead and look at what this all means. And I'm going to jump to back to our idea that in speciation we have a process here which would be called allopatric. So allopatric speciation is probably the most widely cited mechanism for how speciation happens. So I think I defined this last time and suggested that allo means separate, patric means parents, and what they're referring to really is parent populations. So if there is a separation of parent populations that happens, this is the process that initiates allopatric speciation. We have a couple different ways this can happen. We have vicariance, and then we have dispersal. So what vicariance means is it's some sort of a geographical barrier that forms and separates populations. So you can think of like the Central Valley of California. At various times, the California Central Valley, like from what's now Red Bluff or even Redding, all the way down to Bakersfield. Right now it's a valley, like kind of a grassland, woodland valley. At various times, ocean levels have been much higher than they are today. About 30,000 years ago, what is now Redding was underwater. Basically, the Pacific Ocean inundated the valley and covered what's now our area. And during those times, you would have had, say for example, a plant population that existed from the foothills of the Cascades and Sierra Nevada range all the way over to the coast range. So maybe a population of plants that existed in the valley. At times when the ocean levels were higher and the valley was inundated with water, those populations would have been relatively isolated, no longer contiguous. So you have maybe a coast range population and a Cascade range population. During that time of isolation, there is reduced interbreeding and natural selection, sexual selection, genetic drift, mutation, horizontal gene transfer. All those things that happen in one population didn't happen in the other population, such that given enough time, maybe as many as 10,000 generations, if the sea levels receded and the populations were able to overlap again, maybe they wouldn't recognize each other. Sorry. So remember I said here what we're looking at is what causes the transition from microevolution to macroevolution. It's time. You need time, and that's in terms of generations. You need reproductive isolation. So reproductive isolation, where you essentially isolate separate populations from reproduction. And the very first step in allopatric speciation, whether it's vicariance or dispersal, is a geographic isolation. And what geographic means is, in space, you somehow separate the populations so they can't interbreed and exchange genes. And so essentially the gene pools are separated. And they go on their own microevolutionary trajectory, right? So, geographic isolation, according to allopatric speciation, is the first step that will ultimately possibly lead to speciation, right? Okay. So, that being said, during that geographic isolation, which may take as many as 10,000 generations to happen, there's a number of levels of isolation mechanisms that can occur. And what I'm going to do is I'm going to list them out. I'm going to list out these levels of reproductive isolation. So on the exam, there's going to be a couple pages of like brief descriptions, like a short couple sentences, short paragraph with maybe a little picture to help you kind of think about it, that will describe generally a mechanism for reproductive isolation. All these are fair game. There's more on the next page too. But what I want you to do is be thinking about, what is the mechanism that's being kind of described here for isolation. So after geographic isolation, it's obvious. Geographic isolation means, you have an ocean or a mountain range or a glacier that separates populations geographically. Dispersal would be where you have a population that migrates away and colonizes a new habitat. Think of like the Hawaiian Islands. Species had to get there somehow, and they dispersed there. They got there by migration, being carried by something. Okay, so after geographic isolation occurs, so geographic is the first level, I think you can kind of imagine how that would happen. Geographic isolation. Let's go ahead and look at our next levels of isolation. Ecological. So, I guess the way to really define this is, this is a physical isolation that happens independent of the population. Everything I'm going to talk about now requires some level of microevolution to happen. There has to be some change in the population traits for any of these ecological, behavioral, or temporal, and so on to happen, right? Okay, so let's take a look. Here's an example. Here's two species that are relatively similar in terms of their overall kind of body form. So lions and tigers, right? If you look at lions and tigers, these are big cats that occur. I think the lion is mostly in Africa, or today is all in Africa, I guess. At one point in time, lion populations occurred through the Middle East and into India. But now, with human effect, they've pretty much been isolated in kind of savanna areas of Africa. Tigers occur all the way from like the Middle East, or historically from the Middle East, India, and then into Russia, Siberia, and all the way down into like Sumatra, down almost into the Australian island areas. Today, the populations have been drastically reduced to small little patches. But essentially, lions are a savanna grassland species, and tigers are more of like a jungle forest type habitat species. And if you were to show a lion and tiger picture to a third grader, they would clearly tell you, they're obviously different species, right? If they encounter each other in the wild, it's likely they'll kill each other, right? But in a zoo, you can hybridize lions and tigers. The tiglon is a hybrid between a tiger and a lion, and the other alternative is called a liger, ligers. So there's ligers and tiglons, which are hybrids. And the name is based on who the father is, or who the male is. Tiglon is a male tiger and a female lion. And a liger is a male lion and a female tiger. Interestingly enough, the tiglons are sterile, generally speaking, but ligers, which are the male lion and female tigers, are actually fertile in the zoo, and they've perpetuated populations. Like in the zoo in Germany, they have liger populations in the zoo that are potentially 100 years old or so. I mean, so they've gone around for multiple generations, 10 or more generations. So if they produce hybrids that are possibly fertile, why do we consider them separate species still? Yeah. Right. So the zoo is not natural, right? The zoo is an artificial environment. In nature, they don't hybridize. And one mechanism that prevents their hybridization is they occupy very different habitats. There's also other levels that exist as well, like behavioral, and many of these mechanisms could apply to lions and tigers. What I want to emphasize here is we have a specific isolation that's happened. They occupy different habitats, and that's one mechanism that prevents the reproduction. Okay, so the example of ecological isolation here would be like lions versus tigers. I'm using some pretty grand examples here, charismatic megafauna, as they call it. But they occupy different habitats, so that's one reason that they're considered different species. But the basic test is, in nature, they don't hybridize. You don't see ligers and tiglons going around in nature. The idea there is that the hybrid, tigers are very well adapted to their habitat. Lions are very well adapted to their habitat. The tiglons and ligers are not really adapted to any habitat, right? They don't really have the best of all features that exist, all right? Sound good? Cool, pretty simple. Okay, let's go back here. Ecological isolation is how they use their resources. We also have behavioral isolation that is a barrier. So behavioral isolation. Leopard frog populations in the U.S. The leopard frog in general, there's multiple species of leopard frog, but they occupy, most of the southeast, into the south, all the way up into Canada here. And but there are actually several or multiple species that occupy these habitats. And their mechanism for finding a mate is their little frog calls, right? So males will have a song or they'll croak some sort of a pattern. And that croaking song is to attract the females. And in areas where two species overlap or where they coexist, that song is what prevents hybridization. So there is a behavioral mechanism that has happened, potentially during this time of geographic isolation. The frog populations develop independent songs or different mating calls, such that when they come back together again, the call does not attract members of another species, or at least as readily. So it prevents hybridization. So this example would be like mating calls, or song, or plumage or colors, coloration. So another example of isolation would be like in Florida, there are these anole lizards, these little, they're kind of like gecko type lizards in Florida swamps. And in the same area, there'll be multiple species of these little lizards, And how they attract a mate is by using they like kind of inflate their little neck sack, and they use it as kind of like a mating display where the males will, kind of advertise their availability and interest by showing the color of their throat sac. And the females will basically choose the correct species or individuals of the right species to mate with based on that coloration. So if an example talks about some sort of behavior, whether it's a mating call or song or a pheromone or a coloration or a dance, sage grouse do their little dance thing, right? And that's how sage grouse and blue grouse, which can occur in the same habitat, would hybridize, right? Deer, white-tailed deer and black-tailed deer have different behaviors, but maybe mating pheromones or things like that, scents or odors. You get the idea. Behavioral is a major potential mechanism. And you can imagine lions and tigers have very different behaviors too, right? I mean, lions live in female prides with maybe one male, occupying the pride at a time. But there's probably also other behaviors as well that could apply to lions and tigers. But I'm just using, on the example on the test, what I want to encourage you is just use what's available. Don't go off into the world of maybe what could happen. Just use what is listed as the description, and what I'm asking you is to identify whether the description is asking you about an ecological or behavioral example. Okay? Make sense? All right. Okay, and then we have temporal isolation. What temporal refers to is temporal isolation is mating timing. Temporal isolation is where you have the timing of mating, whether it's the time of year, like fall versus spring, or whether it is even the time of day. Some flowers will bloom in the morning and attract pollinators and then close up before noon, whereas other species of flowers will open in the evening. So you can even have temporal isolation that happens in a 24-hour time period, or when the mating calls happen. If it's a timing thing, so anything that has to do with time of day, time of year, time of month, etc., that is an example of temporal isolation. My favorite example for this, which you're not going to see on the test, but I'm going to give you this example because it's a local example, is we have our local Chinook salmon that occur in the Sacramento River. Let me go ahead and just pull this up here. So the Chinook salmon. So our local Sacramento River has these Chinook salmon species, and they what they do is they hatch or they're born in the creeks, rivers and streams in the local area, and then they migrate down the Sacramento River within about a year, and then they spend their basically 1 to 5 years. They spend about 5 years out in the ocean, feeding in the ocean, increasing in size, and then in about 5 years they'll migrate back up to Sacramento River in the Redding area and the tributaries to mate. So the Chinook salmon, there are seasonal runs of Chinook salmon that exist. So temporal isolation is timing of mating or reproduction. So if we go ahead and put a kind of a timeline here, I'm going to start the timeline not in the traditional context of January, February, March. January is considered the first month of the year. Chinook salmon don't really care about our calendar, right? They care about their calendar. And where the Chinook salmon's calendar starts is when the first rainfalls happen that allow them to get up into the creeks to reproduce. And that would be October. So October would be like the beginning of the water cycle or year for these populations. So it's October, November, December, January, February, March, April, May, June, July, August, and September. So if we basically wrote out all 12 months, those are just the first letter of every month, what we have is in the creeks, basically leading up to October, they're kind of hovering around in the river right now, but as soon as the first rain falls, basically, we have what's called the fall run. Okay, and then these are Chinook salmon. Okay, so the fall run, they start migrating up the river in like the end of summer, end of July and August, but they're not mating yet. And right now in September, they're hovering around in the Sacramento River, but they're not mating yet. But as soon as the first rainfall happens, which is usually the end of September or early October, which is why I'm putting little dashes there, but the solid line is basically when their big mating run happens. Okay. All right. So they kind of do their thing from from October, November, maybe into December. This is kind of a general idea. And then there is a winter run that kind of starts in December and goes through, maybe February, March, and potentially into April. There is a winter run. And so they start migrating from the ocean, maybe around Halloween, Thanksgiving time, and they're into the creeks by the new year, by the changeover in what we consider the new year. And that's the winter run salmon. Late fall run salmon and early winter run salmon can hybridize. They'll actually potentially have hybrid offspring here during that time. So fall run and winter run are actually considered to be probably the same species, just kind of varieties of the same species. But then there is a spring run, and the spring run starts, March, April, May, and goes into, June. Kind of the late spring rains, and then eventually what happens is the creeks start drying up, getting a little too warm to support populations because of lack of rain. But we have a spring run. So that spring run is essentially spring through until early summer, and then July, August, and early September is really not any possibility for reproduction. So winter run and spring run overlap, and they can hybridize. So there's hybrids of winter run and spring run. So those are considered, potentially the same species. But there's many fish biologists that would argue that spring run and fall run do not directly reproduce. And if you look at them genetically, like if you go and look at their DNA profiles, they are quite different. They're more different than many mammals or birds that we consider. Much as like a 10 to 15% genetic difference between the spring run and fall run. So it's pretty clear that there's not a lot of gene flow between the spring run and the fall run salmon. And there are many fish biologists that would like to argue that the spring run and fall run should be considered to be separate species because they have no chance of reproducing in nature. But because they hybridize through the winter run, and there might be gene flow that happens over the course of, hundreds or thousands of generations. Right now, there's, kind of some controversy in fish biology as to whether this spring run should be considered a separate species or not. So anyway, but as an example, the overall example here is not to argue whether fall run and spring run are different species from the same species. The argument is that what we have going on is a temporal isolation. There's a potential difference in the timing of mating that happens that potentially would argue that spring run and fall run could be different species. Okay. Questions? All right. So these are all deemed to be what we call pre-mating isolating mechanisms. So these are mechanisms that would essentially result in mating never happening. So these are called pre-mating. Pre-mating isolation. And what that means is there is no potential for mating to happen unless there's some hybridization event that occurs. Got it? Okay, so on to the next level. So on the test, if you see any indication of time of the year, month, time of the day, if time is what the paragraph is describing, you're going to write temporal isolation as the main mechanism of isolation. Okay, we're on a good track here. Let's see where we at. All right. Okay, let's see. Then, if mating does occur, if there's enough of a habitat overlap, if the behaviors are, in line enough that there is an attempt to mate, and if the timing of year is right and mating potentially occurs, we have some of what are called post-mating mechanisms. So these are after mating, but before fertilization. It's called prezygotic because its fertilization doesn't happen. So these are what are called post-mating isolation. And but fertilization is blocked, so it's pre-zygotic or pre-fertilization mechanisms. And there's two mechanisms I want to talk about here: mechanical isolation and gamete isolation. Okay, so for mechanical isolation, insects are a really good example of this. In dragonfly, So in dragonflies, they have a really kind of a peculiar mating process whereby the male will kind of fly around like a suitable habitat, like a creek or a pond or whatever. So the male's flying around. This is the male, the blue one. And in insects, usually the males are more brightly colored because they're trying to attract females. But anyway, what the male does is he has a clasper, or he has little, he has kind of like little forceps or tweezers on the back of his abdomen. And he uses those little forceps or tweezers to grab onto the neck or grab behind the neck of the female. And if you look at, then what happens is once he grabs the female and catches the female reaches around and retrieves the sperm from the base of the male's abdomen. So they don't have like gonad to gonad contact. They basically, the male will have a sperm packet that he puts on his thorax and then catches the female, and then the female retrieves that sperm packet. But anyway, what's going on here is, let's go ahead and jump back here and look at the claspers. These are called claspers in males. And let's see, dragonfly claspers. Logged in on mating here, so let's go ahead and do that. All right, so here we go. This is kind of what they look like. The male's end of its abdomen has these little, what look like tweezers or little pliers, and they're specifically shaped for the species of female that he's trying to catch. So if there's two dragonfly species in the same habitat, the male and female of different species can't mechanically attach. The male can't grab onto the female and hold on to her in the right way. Plus, it's the shape of the claspers that makes the female responsive to picking up the sperm packet. So we have basically is we have dragonfly mating claspers, where the male has to have the right shape of his grabbing mechanism in order to be able to hold on to the female of the same species. So it's kind of like a lock and key type mechanism. You know, it's like you gotta have the right key to get into the lock, and so that's what kind of like these external, mating structures are all about. So that's a mechanical isolation. You have a physical inability to actually mate or transfer sperm. Further, you have gamete isolation. And in gamete isolation, you have egg and sperm proteins. Egg and sperm, there are fertilization proteins. And how this works basically is when you have, say for example, an egg, and then you have sperm cells of the same species. So we have our egg here, and obviously our sperm here. So how the sperm physically attaches to the egg is by having specific mating proteins. There are like receptive proteins on the egg surface. Think of like Velcro, or think of like little docking stations. And then the male has a compatible protein on the tip of his acrosome, which is the very tip of the sperm cell. So how this works then is a male of the correct species will be able to attach to the protein of the egg, because basically the proteins are compatible and they stick. So it's not about like swimming really fast and making a run and go at it, right? It's essentially where the sperm cells will just kind of stick like a magnet onto the egg cell. And once that happens, then the sperm transfer, the chromosome transfer can happen. But if there is a sperm cell of an incompatible organism, like if the different species tries to mate, they will not be able to attach to the egg. And this could be a change as simple as one gene. One nucleotide change, one ATGC DNA code change could result in a sperm cell not being able to bind to an egg. And so this is, matings obviously happen if you have egg and sperm mixing together, but if the egg and sperm can't combine their chromosomes, fertilization can't occur. So this is pre-fertilization, or what the textbook calls zygote, meaning that no fertilization happens. Okay? So, if you see any hint about eggs and sperms or gametes being together but fertilization doesn't happen, it's gamete isolation, right? You got the idea? So again, I'm just kind of sharing from previous tests. I give all these, like, about 8 or so examples on these 2 pages of the test. Just use what is, available at hand. Don't go into the realm of what could happen or possibilities. You're just interpreting the statement as it's written. And so if there's any indication of eggs and sperms not being able to combine, it's gamete isolation. Any indication about the plumbing not being compatible between populations or species, it's mechanical isolation, and so on and so on. I always kind of get baffled at some answers on tests where people bring in things that aren't even described, and so you use the information that's available. Okay, so then we have a whole series of what are called post-zygotic. And post-zygotic, what is zygote? Zygotic. Post-zygotic. What a zygote is it is it's a egg that's been fertilized. Once the egg has been fertilized and you now have a 1-cell embryo that is beginning to divide into 2, 4, 8, going through embryological development, that's called a zygote. Zygote is when you have the chromosomes, the male and female combined, a fertilized egg. You still have mechanisms that could result in isolation post-zygotically. And those mechanisms would be, there's a couple levels of them, but I'm going to go ahead and just kind of combine them all into one. What we're looking at here is we have a hybrid situation where you have 2 species that combine their gametes, eggs and sperm, and you now have an embryo developing, or maybe even a fully mature organism that develops, but there's still isolation. For example, the hybrid embryos might not develop properly. Babies' fertilizations happen, so we've gotten past here, but we could have inviable embryos where the embryos don't actually develop correctly, and it's a kind of like a dud or incomplete birth. Or the hybrids don't survive in nature. This is where the like I said, pythons and ligers, they probably wouldn't survive in either the savanna or the jungle habitat because they're not really adapted to those environments. Or the hybrid adults are sterile or have reduced fertility. So in this case, what we're looking at is hybrid inviability. I'll just go ahead and combine all these together into one basic idea. Is with post-zygotic, we have a hybrid form, a hybrid of 2 different species, but they're inviable. And what that means is they're inviable in terms of development, in terms of their adaptation, and in terms of their sterility. So we're just going to go ahead and kind of combine all these together into the same basic idea is the hybrids just don't work out and they cannot perpetuate themselves. Either they're never born, they don't survive as adults, or they don't reproduce. And probably the most widely known example of this would be horses and donkeys, right? Equines. So you have horses and donkeys. Horses, naturally, their natural habitat or where they're from is kind of the Asiatic steppes, kind of the northern Middle East up into China and Siberia is kind of where the horse habitat is. There still is a naturally occurring truly wild horse, meaning never been domesticated, called Przewalski's wild horse, which occurs kind of in the Russian steppe area. Donkeys, or asses or onagers, they are a natural population that occurred in the Middle East, kind of Egypt and what's now Iran, Iraq, etc., kind of that Middle Eastern region, Syria. And then the other equine species are zebra. There's like 3 species of zebra that occur in Central Africa, mountain regions of Africa, all the way down into Southern Africa. So above currently living equines, there's like about 5 or maybe 6 species of equines. And they all hybridize. A mule is a cross between a horse and a donkey specifically. And actually, a mule specifically is a cross between a male donkey, a jack donkey, and a female horse. That's what technically a mule is. It has to be a jack male donkey and a mare horse for a mule. So let's go ahead and write this example down, and we're looking at for example, a mule. Oops. Let me do this. I'll write this way. So we have a female horse crossed with a what's called a jack donkey, which is a male donkey, and the resulting offspring is what's called a mule. And mules, for the most part, are sterile, right? Most elementary school kids would know that a mule is sterile. Real quick before we do that, does anyone know what it's called if you have the opposite cross? You have a male horse and a female donkey? Anyone know what that's called? So a mule is specifically this cross, where you have a female horse and a male donkey. But if you had a male horse, a stallion, and what's called a jenny, in donkeys, the females are called jennies. I don't know why. But anyway, you know what it's called? It's called a hinny. The term hinny is used to describe the opposite cross of male and female. So it's like our ligers in Taiwan, right? You basically name it based on what the offspring is. Interestingly, you've probably heard that mules are like the best of both worlds, right? They have the smarts and the stamina of a donkey, but they have the physical size of a horse, so it's kind of like a win-win. Whereas a hinny is considered like the worst of both worlds, where you have a small body size, but they are like not very smart. So hinnies are not intentionally bred in captivity because they don't really have the applications. A mule would be like a working animal, whereas a hinny would be like a mistake, right? So anyway, horses and zebras can hybridize, right? You can have a horse and zebra hybrid. Anyone know what it's called? A zorse, right? A zorse. What about a zebra and a donkey? Zonkey. They're called zonkies. Yeah. So anyway, these are all hybrids. So how about like a zonkey? So anyway, so you can have hybrids of these organisms, but what's interesting is why they're sterile. You know, what's the mechanism by which sterility happens? And this isn't always the case, but the mechanism is the number of chromosomes. So horses, I think I'm getting this right. You can correct me if I'm wrong. The chromosome number of a horse is 64 chromosomes. So when I say 2N, What I mean is that their diploid number is 2n is 64 chromosomes. Okay, so where that number 2n comes from is it's the diploid number, it's the combined chromosome number. So for humans, we have 46 chromosomes as our 2n number. And it's kind of confusing because we have things like 23andMe, which is the chromosome testing kits that you can buy. And what they're referring to is really what you have is 23 pairs of chromosomes. You have 23 matching pairs of chromosomes. You got 23 chromosomes from your mom. You got 23 chromosomes from your dad, big dad. And combined together, 23 and 23 make 46. So the 2n chromosome number of the horse is 64. The 2n chromosome number of the donkey is 62. So that means that when the egg is produced, we have, let me just get this done right. We're almost done here. So when an egg is made, this is the egg. The egg has a chromosome number of half that, 32. So you have 1n of 32 chromosomes, meaning it has half the chromosome number of the adult. And the sperm cells of the sperm cell of the donkey has sperm, right? And it has a 1n number of 31. So the idea there is when you combine these together and you make a mule, you basically have a 2n number of 63, right? You add 32 and 31, that's 63. The problem is 63 can't be divided in half evenly, right? You can't get a predictable or consistent number of chromosomes by dividing 63. So basically the eggs and sperms are very, very unlikely to merge with a compatible opportunity. So that's why one mechanism by which the sterility happens is it's an incompatibility of chromosome numbers. That's not really the true full story, but it's good enough for right now, right? So basically the idea, it's a mechanism is you have an incompatibility of the chromosomes that result in the inability to produce eggs or sperms that are compatible. Now, there's usually some horse person in class that'll say there has been examples of specifically a female mule that back crosses with a donkey. There have been examples of female mules that have produced offspring that have matured and basically been a mule. But it's like 1 in 10,000. It's extremely rare. And the basic idea is if there is any isolation, even a reduced fertility, that's considered to be a reproductive barrier, right? Because remember what Darwin said is populations have a great potential for fertility, and any limitation or reduction in fertility is a deleterious or negative effect on that species. So that's the way it goes there. We good? Okay, let's go ahead and take a break, take a little long break because I have to get our stuff out for lab today. We're going to look at the Nigeria, which is the jellyfish, and, hydra, and anemones, etc. So it's not too much of a lab thing, but this is the organism. So 9:15. All right.